Okay, Meredith, tell me, do the slides move, yes or no? Meredith, can you hear me?
Yes, I can.
Are the slides moving?
Yes.
Okay, thank you.
Yep. Did you see my email?
Yeah.
Okay. As I say, I just sent it like 5-10 minutes ago, but.
Mrs. Schaefer has copies of everything. She can send you whatever you need sent to you. So
Okay.
that
Sounds
won't
good.
be a problem.
Okay.
Because they all start with P.
One of mine is actually, so when you download it, really? That's what mine did. For the, it wasn't either the last.
And I don't know why. And I'm like, okay.
Yeah.
Why?
Clearance. Okay. Closing resources to, yeah, okay, whatever. Okay, we will resume. So remember last time we discussed the microstructure of the muscle. We saw that the sarcoplasm, that is the cytoplasm of a muscle cell, is filled with these contractile elements called myofibrils. Myofibrils. And the myofibrils are organized into repeating units called sarcomeres. And the sarcomeres have several different regions in them. So the sarcomere, first of all, goes from a Z disc to another Z disc. That should see. And then in between the Z discs, you have part of an I band. And then a so-called A band. Within the A band, there is this area called the H zone. And then you have another, of course, the rest of the I band. Now, the A zone consists of so-called thick filaments. And as you will see, these thick filaments are composed of myosin. There's several different types of myosins. This one's actually called myosin 2. The I band contains a protein called actin. And those are the so-called thin filaments. Dead center of the sarcomere is a so-called M band, and the M line is actually a site where the myosins attach to them, attach to the center. And there's a protein in the M line called meromycin that helps attach the myosin to the center of the sarcomere. So if we take this structure and we now convert it into a cartoon, we see this a bit more clearly. So here is your H zone. The H zone is really the space between the ends of these thin filaments. The A band, as you can see, constitutes the length of the myosin thick filaments. And they're attached from head to tail, and then from head to tail. So this entire set of thick filaments, a mirror image of each other, directed upon that M line. So these tails all attach here at this M line. That's what meromycin helps you. The thin filaments overlap with the myosin thick filaments, at least partly. And so this I band is really the portion where you only have actin. And that's why it doesn't have nearly the color or the mass that you have with the thick filaments. And if we try to reduce it to lines, we can see here the thin filaments, there's the thick filaments, space in between the ends of the thin filaments, that's the H band. The thick filaments constitute the A band. And then this area here between the ends, if you will, of the thick filaments that just have thin filaments, that's the I band. And if you try to make this more 3D, you can see that it has more of a look like this. And here is something you can't see very well because of there. Makes it look more 3D. Z disc is in fact a disc. It's a 3D structure, as you can see. And here's these myofibrils here, thick filaments here, thin filaments here. That's in cross section. And then you're of course going to have lots of mitochondria and then aspects of the so-called sarcoplasmic reticulum. There's the endoplasmic reticulum in muscle cells, which has been modified to form a structure that may stores calcium. There's your myofibrils, thick and thin filaments. We'll revisit the sarcomere in just a bit. First, let's talk about the innervation of the muscle cell. So the upper motor neurons in the precentral gyrus will send their axon down, and then it will synapse with the cell body of a lower motor neuron, and that lower motor neuron will then extend to a muscle. Now, this extension of the muscle is a bit of a difficult thing, because in order for a muscle cell to be activated, it needs an action potential from will result of an action potential from that motor neuron. How are you going to innervate all these different muscle cells? The axon of that motor branches, and it branches extensively. And it's going to then form a synapse with the motor, with the muscle cell. That synapse is a specialized structure called either motor end plate. It's also called a neuromuscular junction. Very specialized structure. So here is a very beautiful picture of a motor neuron. You can see the axon is very highly branched. You can also see when it synapses with the muscle cell, the connection, essentially it kind of digs to the membrane of a muscle cell. So the muscle cell membrane, the sarcolemma, is highly indented. And then the axon terminus of the motor neuron essentially fills that indentation. And a very intimate association between the muscle cell membrane and the axon terminus. And this is the nature of motor end plate. So here it is in cartoon. And you can see here, again, you have an indentation. Also, the membrane at this motor end plate of the muscle cell, very highly ribbed, very high folded. That increases the surface area that will be in close proximity to this motor neuron terminus. Now, this motor neuron, as you can see, is loaded with synaptic vesicles. those synaptic vesicles are full of acetylcholine. All motor neurons release acetylcholine, all. When an action potential arrives at this motor neuron terminus, voltage-gated calcium channels open, calcium enters, and it induces exocytosis of these synaptic vesicles. These synaptic vesicles will fuse with the membrane and they'll release their payload of acetylcholine, and the synaptic cleft, as you can see here, or neuromuscular cleft, will fill with acetylcholine. The folded sarcolemma is enriched with nicotinic acetylcholine receptors. These are ligand-gated ion channels. They bind acetylcholine, they open. They allow the influx of mainly sodium, also calcium. There'll be a slight efflux of potassium, but that efflux of potassium is pretty minor. This will cause depolarization of the membrane. This depolarization, however, is local. So it turns out the resting potential of a muscle cell is about 100 millivolts, thereabouts. This depolarization will increase the membrane potential to about minus 60 millivolts. That is threshold for voltage-gated sodium channels that are found throughout the membrane of the sarcolemma. That is going to depolarize the membrane of the sarcolemma. That's going to have pretty profound implications for the function of the muscle when it comes to contraction. We're going to leave that there for just a bit. Okay, we talked about that already, the branching of the motor neuron axon. The motor neuron will innervate several muscle cells because of the branching of the axon. All the muscle cells that are innervated by the motor neuron plus the motor neuron itself, that collection is called a motor unit. A motor unit. On the average, it's going to depend on the muscle, but on the average, a motor neuron is going to innervate about 150 skeletal muscle fibers. It depends upon the precision required of the muscle. So the lower motor neurons that innervate the muscles of the eye, there you want less force, more precision. Those will innervate about 10, 15 motor muscle fibers apiece. The biceps brachii, however, which is a flexor but it's also a pronator. Those lower motor neurons will innervate about 2,000 fibers apiece. And the gastrocnemius, it's even higher. So if I ask you on a test, how many muscle fibers does a motor neuron innervate? The answer is, it depends, because it varies from muscle to muscle, doesn't it? But here's a rule you can bank, and that rule basically says the more precision and less force, the lower the number of muscle cells that motor neuron will innervate. The more strength you require, the greater the force, the higher the number. Now, and this is a picture from your book simply showing the innervation of all these muscles. So the muscles, motor neuron, that's your motor unit. This, that's your motor unit. Now, let's think about this from a clinical perspective. Polio virus infects the GI tract. It then uses the nerves that innervate the GI tract. It follows up those axons in a retrograde fashion. It gets eventually to the spinal cord and it attacks motor neurons. It can destroy those motor neurons. If it destroys those motor neurons, what happens to your ability to contract that motor unit? It disappears. And if that happens to be muscles in the lower part of your body, you lose the ability to walk. If it's just a few of these, then typically, if you're young, what might happen that some of these other nearby motor neurons will start to extend collateral axons to try and innervate those. Now, as you age, however, some of the branches can start to die off. And then you're left with a so-called post-polio paralysis that can occur decades after you had polio and went through polio and even learned to walk again. So this is a consequence essentially of the death of those neurons. Also, and here's a neuromuscular junction. We talked before about a immunological disease where you make antibodies to nicotinic acetylcholine receptors. Remember what that genetic disease was called? Something gravis? Yeah, you got one part. Myasthenia gravis. Myasthenia refers to muscle weakness. Myasthenia gravis. In that case, you destroy the elements of the neuromuscular junction. Start to destroy actually elements of the muscle cell. That can regenerate, but as you keep damaging this, eventually the motor neuron is going to crack. Now, think about muscles. The muscle and the motor neuron are constantly talking to each other. The motor neuron actually releases small little peptides called neurotrophins. The muscle needs those neurotrophins to survive. When you kill off a motor neuron, like in a spinal cord injury, these muscles will start to atrophy. And they will shrink in size. Your body will now start to, there's all that great protein reservoir there. It'll use it. And the blood vessel vasculature even starts to retract and what have you, because it doesn't get the neurotrophins. You could see why trying to fix spinal cord injuries is such a difficult thing, because it's more than just getting the nerves there. The muscles themselves now actually need to be regenerated as well. So this innervation ratio, that is the proportion of the muscle cells that are innervated relative to per motor neuron. And we talked about the ratios here. That innervation ratio for big muscles is very high. Smaller, more precise muscles, it's very low. All right. Skeletal muscles have sarcomeres, so do cardiac muscles. Smooth muscles don't. Smooth muscle contraction is very different than it is with cardiac and skeletal muscles. Contraction of smooth muscle tends to change cell shape, whereas cardiac and skeletal muscles will actually shorten. All right, let's get back to the myofibrils. So with the myofibril, here's a nice transmission electron microscope of a striated muscle. The striations are due to the sarcomeres. You blow this up. Here is your zebra stripes. There's your A bands. There's your, who's that? I bands, A bands, I bands, A band, I band, A band, I band, so on and so forth. And then blow that up even more. Here's your M line, A band, I band. There's your Z disc. And we go back to this picture. And remember that this part of the sarcomere is a mirror image of this part of the sarcomere. So what happens to one side will also happen to the other side. Keep that in mind. Very good. Here's what happens when muscles contract. When muscles contract, the light, thin filaments get pulled together. So what happens to that H zone? The H zone disappears, and at full contraction, the I band will also disappear. So my exceedingly mnemonic for remembering this is that muscles, when you look at them, say high, but the high disappears when the muscle is fully contracted. The H zone and the I band disappear by full contraction. And you can see this here. This Z bands are getting pulled together. Here's a Z band, here's a Z band. This one doesn't move. At contraction, here's the I band. Let it, look at it. Do you see it now? No. As at full contraction, there's no high anymore. Here's your H zone, smaller, disappeared. There's no high. So the contraction takes away the high. The ends of the thin filaments are pulled together. They're pulled together. You can see this in a cartoon like this. Ends of the thin filaments, you pull together, and now that H zone goes away, so does the I zone. So on contraction, H zone and I zone will shrink. At full contraction, they disappear completely. The ends of the thin filaments slide over the thick filaments and are pulled together. So if we use essentially this smart art and say fully contracted, uncontracted, we can then go from uncontracted again, mirror image, comes to one side, happens to the other. We then start to contract and slide over the thick filaments here like this, as you can see. They'll slide toward each other. They get closer and closer together. That I band gets smaller and smaller. H zone gets smaller and smaller. The I band, however, does not narrow. Excuse me, the A band does not narrow, because what's the A band? The thick filaments. Thick filaments aren't moving. They're causing the thin filaments to move. And then at full contraction, there's no high anymore. The muscle no longer says hi. Okay, questions about muscle microstructure, questions about contraction. Meredith, again, if you have any questions, please unmute yourself in something, because if you send a chat, I won't see it.
Okay.
Okay. Man, why is she so loud? Okay. Okay. Now let's look at the sliding filament theory of contraction. The myosin comes in two parts. There's a head and a tail. A head and a tail. Myosin, some myosins don't work as dimers. 1, 2 does, which is one of the reasons why I have myosin 2. Well, actually, it's called myosin 2 because it's the second myosin to be characterized in detail. But it works as a dimer. The tail is responsible for dimerization. The tail essentially will do this. They also meet each other and then they'll wrap around each other. And they're going to secure each other really well. And now the heads are free to do their dirty work, essentially. It's like that. So here's your head, here's your tail. This is wrapped kind of like a caduceus. You know what I mean when I say caduceus? The symbol, medical symbol, the snake around the stick. It's like a caduceus. It's wrapped like a caduceus, and then here's the heads ready to do whatever they do. Okay? Now, the head is a molecular machine. When it hydrolyzes ATP, it changes its conformation. It's able to cock and grab, cock, pull, because of the hydrolysis of ATP. Proteins that are able to change are able to move like that because of hydrolysis of ATP are called molecular machines. Myosin is a molecular machine. And that simply summarizes from your book what goes on with contraction. Let's look at the cycle now. So, myosin, as you can see, when it is bound to ATP, essentially is detached from the actin thin filament. When it hydrolyzes ATP, it now goes from this kind of fully cocked sort of structure to a more open cocked structure. So it goes from fully cocked to, I don't know, half cocked, I guess, after it hydrolyzes the ATP, but it's still bound to the ADP and phosphate. Now, the myosin head has on it, in addition to ATP binding sites, it has a receptor actin, has an actin binding site. And when it releases the phosphate, the myosin will bind to the actin. So it goes from here, binds ATP, more of a relaxed cocked, spits out the phosphate, binds to the actin. Then it's going to release the ADP is going to perform something called a power stroke. So cocked, uncocked, binds, and then it pulls the actin like that. And when it binds ATP, it then releases itself from the actin. So here, power stroke, and then it binds and it gets released from the actin. Hydrolyzes it, cocks it again, spits out the phosphate, binds the actin, spits out the ADP, pulls, binds ATP, release. Hydrolyze, spits out the phosphate, spits out ADP, power stroke, binds ATP again, release. Hydrolyze, and there it goes the cycle again and again and again. This is how mucin pulls the actin thin filaments together. Keeps binding, pulling, releasing, binding, pulling, releasing, binding, pulling, releasing, binding, pulling, releasing. Now, when you die, you don't make ATP anymore. So myosin binds and it can't let go. So the muscle gets really stiff. What do we call that? Rigor mortis. And it's explained by the physiology of myosin and actin.
Is it possible to like deplete your ATP so much that even when you're alive, that happens?
Yeah. Yeah. There was an Olympic runner. You guys are way too young to remember this. What was she running? She was running, I think, the 10, was it the 10? I think she was doing 10K. She was running 10K and she had to not eat before meets because she got very nervous. She'd up it all. And if she upchucked, she'd lose a lot of electrolytes. So she just didn't eat. Her race was in the morning and she didn't eat. And then the race got delayed. So she couldn't eat. And then it was time for the race. So she downed a bunch of water. And so on an empty stomach and 10K, I mean, that's a grueling race. Grueling race. She got to the last lap and she got, I think it was in something like 50 yards of the finish line, and her body completely shut down. And she stiff and literally like that. And the techs ran to help her, and they were trying to get her to move. Her muscles wouldn't move. She stiffed up like she had no ATP left in her body. She had nothing to burn, essentially. So they got an IV in her to get her glucose, and then she was able to relax. But she was in this horrific pain because her muscles were stiff like that, and I mean that's gotta hurt.
If you force the muscles to move, can you do damage to them?
You might tear something, yeah, potentially. Once they got the glucose in her, they were able then to do that, but they had to put the V in. And one of the, there was an emergency doctor on hand who was specialized in athletes, and he knew exactly what had happened because he'd seen it before. So he said, get D5W in her right now. Because they're going to do saline, because that was she's dehydrated. Well, she's glucose, so D5W, put D5W in her. And they had her on her stretcher, and literally she was loosening up already. But she's screaming, and it was just, it was hard to watch. Cramp action is due to damage to the muscle because you've overdone it, and it's leaking potassium, and the muscle starts to get kind of potassium damage because of the damage to the muscle. It's usually that happened to me once in high school. I was playing football, and when your nose guard, virtually every day somebody rolls on your feet, your legs, and because I'm not very tall, I was bottom of the pile, so I was constantly getting stepped on and what have you. And it was third quarter. It was towards the end. Yeah, it was towards the end of the third quarter, and it was down, set, and I just I can't move, and I just I shoved it as much as I could because if I shoved it, the pain stopped, and then every time we went down, it was like, ow, 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 ow. And next time we were off the field, the trainer sitting there pounding on my calf, trying to get the knot out. And it was damaged so much because people kept stepping on me. And there's this one guy, just every play, he just makes sure to step on me. I'm getting so mad. It's ready to, high school, you're full of hormones and you're stupid and you're ready to take a swing at somebody to show them what a man you are. But that happened to me, and that was cramped for the next day. Literally, I was walking and I had to work at my dad's packing house the next day, and I was pulling my leg, and it was awful. Tetanus, yeah, tetanus. Now tetanus goes away because tetanus is due to hyperactivation. Yeah, tetany. No, tetanus is caused by tetanus toxin. That is due to a toxin that prevents the muscle from relaxing. So it cramps up and then it spasms, and spasms are very powerful, very painful. But tetany that you induce in the laboratory, that can happen also through hyperactivation. Yeah, but that tetanus goes away pretty fast. Now, in a not activated muscle that's not activated by a motor neuron, the actin is not available to the myosin and here. It's covered. The myosin binding sites on the actin is covered by this long filamentous protein called tropomyosin. Tropomyosin from the heart is a marker for a heart attack. So cardiac tropomyosin will usually appear soon after a heart attack. It's one of the markers for a heart attack. Skeletal tropomyosin is not a good indicator for skeletal muscle. Creatine kinase is a much better marker for skeletal muscle damage. So this tropomyosin has to be moved. And you can see it is associated with a 3-polypeptide complex called troponin. And troponin is also very sensitive to calcium. Now, when troponin is by calcium, it changes conformation. And when it changes conformation, it essentially drags tropomyosin off the parts of the actin that are bound by myosin. So now myosin can recognize it. So here, as you can see, calcium floods the cell. Now the muscle can contract. Without calcium, even if you have ATP, the myosin can't bind the actin. You can't contract the muscle. So here's again this smart art that shows you the way this works. You have the tropomyosin covering the actin, the parts of the actin at least that bind to the myosin. Here, designated by kind of olives, and the whole of it is the part that binds the myosin. Myosin is bound by ADP and phosphorus, phosphate. It's cocked only when at calcium because of the activity of the motor neuron binding acetylcholine and so on and so forth. Do you have the relinks of calcium? Calcium will bind to troponin, that drags tropomyosin off the sites now that bind acid. Now myosin can bind the actin, and it does, then start to cycle. It will release the phosphate, it'll bind, and then it releases the ADP. There's the power stroke, and now you start to pull the thin filament towards the other end of this thin filament, and you contract. Then it's bound by ATP, it gets released again, and so on and so forth. Blah, 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 blah. And when the muscle stops receiving signals from the motor neuron, calcium then is returned to the sarcoplasmic reticulum, and then troponin will return to a native structure Rapamycin will now cover the actin again. And that's the contraction cycle. Okay. So let's watch a video. And I don't know if you'll be able to see this. I'm going to try and see if you can see this. Let me. Okay. Need to turn this on, and then oh, stop you. Stop. Okay. Now, let me see if I can get Meredith to see this. If I share this. Okay. All right. I should be able to see that. Hopefully she can hear that. And there we go.
During contraction of a muscle, calcium ion binds to troponin. This moves tropomyosin out of the way and uncovers binding sites for myosin on the actin myofilaments. ADP and phosphate are attached to the myosin head from the previous cycle of movement. The myosin head attached to the exposed binding site on the actin myofilament to form cross bridges, and the phosphate is released. Energy stored in the head of the myosin myofilament is used to move the head. This causes the actin myofilament to bypass the myosin myofilament. The ADP is released from the myosin head as it moves. The function of the actin and the myosin head is broken when the ADP module binds to the myosin head. The ADP is broken down to energy, which is stored in the myosin head, will be used for movement. The head of the molecule returns to its upright position and is ready to bind to actin again. If calcium ions are still present, the entire sequence is repeated.
All right. Now let's get rid of that infernal buzz and then go back to our okay. All right. Now, from whence comes the calcium? This is the part that's a little complicated, but we'll go slowly and try to get everybody on the same page here. Skeletal muscles are unusual in that they have these structures that are indentations of the membrane that go all the way through the cell and are called T tubules or transverse tubules. T tubules or transverse tubules. These T tubules run parallel to the sarcoplasmic reticulum. So if you look at this picture here, you'll notice the sarcoplasmic reticulum forms a web-like filigree across the cell, but it has these inflations. These are called terminal cisternae, and these terminal cisternae virtually always are very intimately associated with these transverse tubules. Now, why is this the case? This is the case for this reason. When you get an action potential, when you depolarize this membrane, that depolarization will spread to the T-tubule. Because remember, the T-tubule is part of the membrane. So you depolarize the membrane, you get it to threshold, you open voltage-gated sodium and calcium channels. That depolarization will now be felt in the T-tubule, and there's a signal that's sent to the terminal cisternae as well. What does that signal look like? It looks like this. There's a bit of cell biology here. Don't let that overwhelm you. Just go with me here. So here we have nicotinic acetylcholine receptors. Those are ligand-gated channels. Those only give you a local depolarization. But that local depolarization that you get at the membrane that constitutes a neuromuscular junction is very powerful. And it spreads because in that neuromuscular junction membrane and nearby, you have voltage-gated sodium channels. You get depolarization in the muscular junction that spreads because of these voltage-gated sodium channels. Now, look where the voltage-gated sodium channels are located. They're also located in the T-tubule. So the depolarization starts here, goes here, and spreads here. Therefore, when you get a depolarization in this membrane, it will inexorably spread to the T-tubule. All right, you with me? Now, in the T-tubule, you also have voltage-gated calcium channels. But these voltage-gated cal channels are bound together with a release channel that's found in sarcoplasmic reticulum. These two are physically linked together. This channel is also called a ryanodine channel. Ryanodine channel. And it's called the ryanodine channel sometimes because the small molecule ryanodine binds them and causes them to open. But with these channels, when calcium, voltage-gated calcium channels are depolarized and open, these release channels open as well. So these open, so do these. And now all this calcium stored within the sarcoplasmic reticulum floods the sarcoplasm. It floods the sarcoplasm. That calcium now binds to the troponin. Troponin changes shape, moves the tropomyosin off the actin. So now the myosin can bind it. The ATP from the mitochondria will cause the myosin now to undergo its power stroke cycle, and you'll drag the thin filaments toward each other. So here's this smart art. You get a local depolarization at the motor end plate membrane. That then spreads because of voltage-gated sodium channels. Those voltage-gated sodium channels, the action potential, the depolarization that they promulgate will go to the tubule. From the T-tubule, it goes to the terminal cisternae, and that causes the calcium release channels to open because you've also activated these voltage-gated calcium channels. The opening of the release channel, ryanodine channels, floods the cytosol with calcium. Calcium now binds to troponin. It drags tropomyosin off the myosin binding site to the actin, and now myosin will be able to bind to actin and carry out contraction, just like you see here. And once again, we have a video to show you. So I need to go through this rigmarole, try and show this. Okay, let's pause you. And then let's try and get Meredith to be able to see this. All right. I'll show you that. Okay, we'll restart this, turn this back on, and then try this again.
An action potential introduced at the neuromuscular junction is propagated along the sarcolemma of the skeletal muscle. At the T-tubule, the action potential is propagated on the T-tubule membrane into the interior of the muscle cell near the end of the sarcoplasmic reticulum. The depolarization of the T-tubule membrane causes nearby voltage-gated calcium ion channels on the sarcoplasmic reticulum to open, resulting increase in the permeability of the sarcoplasmic reticulum to calcium ions. Calcium ions then diffuse from the sarcoplasmic reticulum into the sarcoplasm. Long momentous tropomyosin proteins lie on two sides of the actin myofilament, covering up the sites where myosin can bind to the actin filament. Globular troponin proteins are attached to the tropomyosin. When calcium ions bind to troponin, this causes troponin to change conformation and move the tropomyosin. When the tropomyosin moves, this uncovers the myosin-binding sites on the actin, allowing myosin heads to bind and form cross bridges. The cross bridges play a role in muscle contraction.
Okay, so let's go back to there. Okay, and now, and that's the end of that slideshow. So